The mobile phase reservoir holds a solvent which is referred to as the mobile phase because it moves. There are usually a minimum of 2 reservoirs in a system. There are 4 lines in the degasser which go into the vacuum. It takes the bubbles and entrapped gases out of the solvent before going into the pump. If gas was to be present in the eluent, this would be detected as noise and would cause an unstable baseline. The pump is used to generate and monitor a specified flow rate of mobile phase, typically milliliters per minute. The sample tray holds the vials and rotates to allow the injection syringe to collect the samples. The injector is able to introduce a specific amount of the sample into a continuously flowing mobile phase stream that carries the sample into the HPLC column. The separation is performed inside of the column. The column contains the chromatographic packed material needed to effect the separation. The packing material is called stationary phase because it is held in place by the column hardware. The detector is needed to see the separated compound bands as they elute from the high pressure column. The information is sent from the detector to a computer which generates the chromatogram. The mobile phase exits the detector and is either sent to waste or collected as desired.
HPLC computer software.
This is a software to perform the HPLC. It is called Empower Pro. To begin the procedure, we click Run Samples. This screen is where we instruct the machine what to do. Under the heading The Function, we click the drop-down menu and select Inject Samples. Before running the samples, a number of steps must be carried out to prepare the HPLC instrument. This screen shows the startup procedure for the experiment. Direct function is selected and the following conditions are carried out. A function called wet prime is carried out for 10 minutes in order to pass the new solvent that will be used through the system. This helps ensure there is no residue from previous experiments remaining in the system. This is followed by a function called purge injector. This is where a degassed solvent is passed through the tubing as far as the column's connection in order to drive out previous solvents and air from the system. Every HPLC has a valve for this purpose which diverts the solvent out in between the pump and the injector. The final function carried out before injecting the samples is the condition column. This is used to prepare the column for the experiment. When a column is connected for the first time or after it has been stored for a long time, it needs to be washed with the storage solvent to get wetted. The solvent should be allowed to elute freely for 10 minutes to release possible dust particles, any residue that may be present, or even air. After these functions are carried out, the sample can be injected into the system. Here the shutdown method is set up in order to tell the device to stop running after all the samples have been run through and the results obtained. The shutdown method is very important in order to stop the machine running unnecessarily. If the system continues running, the solvent, which is the mobile phase, will eventually run dry, which will result in damage being caused to the column and causing the system to become airlocked. In the flow section of the instrument method editor, a number of different factors can be changed. The pressure limit is set at 4000 for the high limit and 0 for the low limit. The pump mode is set at isocratic conditions and a speed of 2 mL per minute. The total flow is set as 1 and is made of 100% solvent A. In the temperature section, the column temperature and the sample temperature can be set. The HPLC instrument is capable of altering the temperature of both. This screen shows the contents under the solvent section. Here you can name the solvents used in the procedure. Under the settings for the PDA detector, the range in which the results will be produced is selected as well as the resolution. The sample rate and the filter time constant can also be changed here. The content is then saved under a suitable title, and when the HPLC instrument is ready, it will start the experiment. When the experiment has been completed, to access the results, click on the Browse Project option on the software's main screen. Here there will be a number of sample sets. Right-click on the sample set you have previously done, then click on the Review option to have a look at it. This screen shows the final peaks of the samples being tested. The first graph is the 3D contour plot, which is specific to the PDA detector. This is where you can alter the wavelength. There is a table at the bottom of the screen which holds the information for the peaks such as retention time, area, percentage area, start time and end time. All this information can be used to calculate the concentration of the unknown sample along with the percentage discrepancy. This is the advantage of using this HPLC software.